cortical granules. Do you remember what other cell type we talked about required calcium ions for vesicles to fuse with the plasma membrane? Not this semester, but in Bio430. What event required calcium ions for fusion of vesicles?
Neurotransmitters.
Pardon?
Neurotransmitters.
Yes. So the synaptic vesicles fusing with the presynaptic membrane required voltage-gated channels of calcium to open. Well, here we've got a voltage current created. All right. So not only does that change the shape of the integrin, but it results in the slow block of polyspermine, which occurs as a result of, and I'm getting close to my line here. So a slow block for polyspermine. Can I erase this description of a fast block? All right. Since I'm limited to my cell here. And that is a release of calcium ions, or an entrance via the plasma membrane. And that results in fusion of these cortical granules. And their contents move into the space between the egg plasma membrane and the zona pellucida. And two things happen. The zona pellucida hardens, and the zona pellucida moves away from the plasma membrane, so there's an increase in the space between the zona pellucida and the egg membrane. Now, in the sea urchin, there is not a zona pellucida, but there is what we call a periavitelline membrane. And so in fertilization through the microscope, you can actually see this membrane move out. And so let's watch a video of that. This image right here is that showing that from the sea urchin. So these are the cortical granules. All right, they fuse with the plasma membrane. Their contents move into that space, and it separates. That kind of functions to pop off any sperm that are already bound to the integrin molecule. So as it hardens and moves out, any sperm that have already previously bound to integrin before the fast block are knocked off. This is using fluorescent and phase contrast microscopy to show the cortical granule. You can see
a sea urchin egg during fertilization is visualized here simultaneously by phase contrast microscopy and by fluorescence microscopy. The egg contains a fluorescent dye that becomes brighter in the presence of calcium ions. When a sperm cell fuses with the egg, the fluorescence image shows a wave of calcium ions that sweeps through the cytosol.
The arrows are not in the
initial
cell.
point of sperm-egg fusion. Following the path of the calcium wave, we see a membrane called the fertilization envelope rising from the cell surface. The fertilization envelope protects the fertilized egg from the outside environment.
That would be the
and
zona
prevents the
pellucida in
entry of
humans.
additional sperm. The rise in cytosolic calcium triggers an elevation of the fertilization envelope through the process of exocytosis. Exocytosis releases hydrolytic enzymes stored in vesicles. Action of the released hydrolases causes a swelling of material surrounding the cell, which in turn elevates the fertilization envelope. Exocytosis can be visualized directly in this system. For this purpose, the plasma membrane is labeled with a fluorescent dye, seen on the right. Each time a vesicle fuses, it leaves a depression in the plasma membrane, which, in the optical sections shown, appears as a ring of increased fluorescent staining. On the left, differential interference contrast microscopy is used to directly view the exocytic vesicles that underlie the plasma membrane. The vesicles are visible here because they are densely packed with protein and consequently have a different refractive index from the surrounding material. Each time a vesicle exocytoses, it disperses its contents and disappears from the image.
So I want to show the
This effect
fertilization
is best seen
again.
when we step back and forth between adjacent frames of the movie.
Tengo sueño.
phase contrast microscopy and by fluorescence microscopy. The egg contains a fluorescent dye that becomes brighter in the presence of calcium ions. When a sperm cell fuses with the egg, the fluorescence image shows a wave of calcium ions that sweeps through the cytosol, starting from the initial point of sperm-egg fusion. Following the path of the calcium wave, we see a membrane called the fertilization envelope rising from the cell surface.
You can actually see the male
fertilization
nucleus right there.
protects the fertilized egg from the outside environment and prevents the entry of additional sperm. The rise in cytosolic calcium triggers an elevation of the fertilization envelope through the process.
All right, so in this image, you can see that we have an oocyte because we have the DNA fusion of the sperm with that integrin molecule. Now it's caused the cell to undergo its second meiotic division. So this would now be an oocyte. And we have the first polar body, which is what chromosomal pattern?
N.
First polar body.
2N.
2 parentheses N. And now our oocyte is just N. And our second polar body is just N. All three, though, are haploid cells. Remember, the secondary oocyte and the first polar body are haploid cells. We just happen to have 2 copies of each haploid chromosome. So now we move into the formation of the zygote as the relationship between the male pronucleus and the female pronucleus occur. And they line up in the first metaphase of mitosis. Meiosis is not going to occur again until these cells become gametes. All right, so if they are sperm cells, this is a male, or if this is a female, when their spermatogony or oogony are formed and move into primary or secondary oocytes, then meiosis will occur again. But from now on, all of these cell divisions are going to be mitotic cell divisions. So still within the zona pellucida, we have the N chromosomal material of the sperm and the N chromosomal material of our oocyte, if you will, because here's our polar bodies, migrate towards each other, replicate, line up along the equatorial plate for metaphase, and complete the first mitotic division. We call this cell, as it's approaching and now they're associated with each other, as zygotes. All right, so that's what we identify the first cell, the oocyte fertilized with the egg, is a zygote. Yes?
You said as they're approaching each other, is a zygote? Okay.
Right. Well, they have to be, just out here and out here, it's kind of still the oocyte, which is an egg nucleus, which is the sperm nucleus. But then when they start replicating and start the steps of mitosis, we can consider them to be one cell.
Okay.
That zona pellucida, by the way, stays around all of these cells as they continue to undergo mitosis until they're found within the uterine cavity. All right, and then it breaks down and it's a process known as hatching. So notice the size of the cells as cell division occurs. With the zona pellucida, that's not going to get any bigger. Instead, as we go from 2 cell to 4 cell to whatever, 6, 8, 16, 32, each cell gets smaller and smaller. However, its genetic information is going to be the same as that original zygote, because all of these are just daughter cells of that original zygote. They, however, have a different environment. So what causes some of these cells to become the embryo, some to become the amnion, some to become the placenta? And some of the placental cells will form the true placenta, some will form a non-functional placenta that doesn't develop any branches associated with blood vessels.
Does it have to do with the zona pellucida?
Well, that's part of it, and part of it is if you look at the 4-cell stage, you know those things you can fold out of paper? You put your fingers inside and you can open them and close them different ways. All right, so they are kind of shaped like triangles. So imagine those cells as pyramids, and we have one cell out here that's exposed to the surface of the zona pellucida and the fluid there. And that part of the cell is polar because The other part of the cell is towards the center of this ball and is in contact with 3 other cells around it, whereas this component is not. So we've already created a different environment, even in the 2-cell stage. We have that part of the 2 cells that are approaching each other versus away from each other. As we get to the morula, about 3 days after fertilization, it's a solid ball of cells. But if we look at that in cross-section, we have cells in the center of that sphere and then cells that are out on the edge. Again, a different environment, a different concentration of chemicals produced by these cells or underneath the zona pellucida. And that contributes to different inducing factors and growth factors that are affecting their changes. One of the most interesting experiments in development I read from a paper when I was in graduate school, 80. And an experiment, chicks have a developing period of about, it's not gestations or not in the uterus, but of about, I think it's 21, 23 days. And in this experiment, they had taken the shell off of the egg, opened up the membrane, and with a sharp scalpel, excised a small component, less than a millimeter cube, out of what would become the wing area, the bud area. There were no buds, limb buds visible yet. And then made an incision along the back of the dorsal surface of the embryo and put that tissue inside it. And then put the shell back over. And when the chick hatched, it looked perfectly normal. Then they waited just a few hours, 2 or 3 hours, and repeated that experiment. And this time when the chick hatched, it had a wing growing out of its back. In that short period of time, 2.5 hours, the cells had already been committed. They weren't committed before, but they were already committed to which of that genetic material was going to be expressed in the development of the bones and the muscle tissue of a limb that were absent in the development of a back. Okay? We see that same thing when we see the outgrowth of the optic nerve, inducing the surface epithelium of the embryo to invaginate and form the lens. Well, this goes on. It's just this complicated arrangement and concentration of chemicals where the cells are affecting each other. And so that's starting to happen already right here. One cell in one place is not in the same environment as a cell somewhere else in that ball. So in this pattern, in this diagram here, you can see the secondary oocyte being ovulated here, picked up by the fimbriae, going through the infundibulum. Fertilization typically occurs in the ampulla, and then it starts its cell division, again still surrounded by the zona pellucida, and it moves down the oviduct. So the morula, the solid ball of multiple cells, day 3. Day 6, it moves into the uterine cavity. And then if the uterine surface is ready for it, it will settle onto usually the posterior aspect of the uterine cavity and start to settle into the stratum functionalis. And that usually takes place between days 7, 8, 9. This is 7 through 10. It's typically days 7, 8, or 9. So within a week after ovulation, okay, give or take a day, this embryo structure, blastula structure, is implanting itself in the uterine wall. So where are we on our menstrual cycle when we look at that? Remember, day 1 is the first day of menses. Day 14 is ovulation. So fertilization has to occur within the first 24 hours of that period. By day 3 is the morula. So this would be the end of the 3rd week, 7 days after ovulation and fertilization. So implantation is going to occur somewhere in this span. What's happening? What's the hormone that maintains the vascular integrity of the stratum functionalis? Progesterone. All right, so if fertilization doesn't occur, we typically see progesterone starting to fall at about the time, it's a day or so after implantation would normally occur. So there goes our implantation. So the fertilized egg and zygote morula cells are releasing a hormone which is typically the one that's tested for with monoclonal antibodies for pregnancy tests, known as human chorionic gonadotropin. That maintains the corpus luteum, so that progesterone levels remain at a level high enough that the stratum functionalis is not shed. So the blastula is intimately involved in maintaining its own survival. So here's a video of the cell changes divisions of days 1 through 5.
It was so soothing.
Who wants to fall in sleep? So over there on the far left is a little impression that would be the male pronucleus. You can see a polar body, just real briefly up there on the upper left as well. There it is again.
Oh, wow.
So polar bodies are trapped. They're still with this tissue, and the zona pellucida is still intact. It's not until the hollow ball of cells, known as a blastula or blastocyst, hatches within the uterine cavity, and that zona pellucida is gone, that those polar bodies are released. All right, so we're going to start talking about implantation. So the settling of the blastocyst